we go ahead and get started? As you guys all know, Allie is co-advised by Bruce, Maggie, and myself, so we're going to try to do a joint introduction. We'll see how that goes. We weren't able to choreograph it much, but...
at all. No choreography at all.
So the day is finally here. Allie's been here for 6 years. She's been kind of the foundation of both of our labs. And she came to us from University of Chicago, where she got her bachelor's degree with two really well-known marine ecologists. So she came in with a lot of experience and a lot of drive and interest. And in particular, in understanding community ecology, and in particular marine community ecology with macrophytes, seaweeds, seagrasses. And I remember the first time that I sort of sat down with Allie and talked to her about what she wanted to work on. She was like, I want to do, really, really good theoretical community ecology. I want to study species interactions, and I want to understand how these communities are going to change with climate change. And as you'll see, she's basically done all of those things. So, her talk will speak for itself. Despite this picture that looks like Allie doesn't want to get her feet wet, and that she thinks tide pools look disgusting. She actually is really an incredible field person, scientist. She's, pretty small, but carries around packs that weigh like 100 pounds and like jumping over rocks and just just like barreling through the intertidal, making sure she gets all the stuff that she needs to get done in time. And in many ways, that's kind of the most impressive part of what she's done, is all the field work that she's done. So Bruce, you want to talk? Bruce is the field guy, so you can talk a little bit more about that.
We're all field guys. So, yeah, she jumped right in when she arrived here, and has, as you will see, done a tremendous amount of really interesting stuff aimed at both empirical understanding and theory. Allie was very enthusiastic, so her first summer here, she volunteered to come out and help with us set up an experiment that we had going. We've been running it since 2005. And a little background, we have something going on in our lab that's called the Wall of Shame. I don't know if you've heard of it. It's a way of highlighting people's screw-ups. And just as a word of full disclosure, I have the most screw-ups on the Wall of Shame. I have the biggest.
But they're not on the wall, right?
Well, many of them are, not all of them are. So with another new person in the lab, Allie came out to a site called Yahat Beach, and we explained to her how the experiment was set up. It was coated with washers with stainless steel numbers and letters pressed into them. And we told her, okay, the C stands for control, the R stands for removal, the I stands for let the algae come in, or the inverts come in, and the A stands for let the algae come in. And so she and the other person started off in this one plot, and they busily scraping away. You're supposed to take, the inverts or the algae out of certain plots. But she had gotten confused, and she thought C meant clear. Which she did. So that part of our experiment got restarted. And she had her first entry on the Wall of Shame. But after that interesting start, everything was great. She only got on the Wall of Shame a few more times. And let's move on.
Yeah, I also wanted to just mention that Allie also did a lot of teaching, and she taught in Bi 450. She's an incredible TA, and I just want to give her credit for that. I think she's going to be an excellent teacher going forward, and she really, made an impression on undergrads and really kind of helped inspire them, and I think she's also done that with new grad students as well. So here she is in the field, looking terrific.
Expounding.
So with that, let's have Allie come on up, give your talk, and we're all looking forward to hearing what you have to say.
Yes. Okay. Okay, well, thanks everybody for coming, and thanks for the introduction. It's very kind. I'm going to talk to you about my PhD research today, which, as has been mentioned, is in the Rocky intertidal. But I'm going to start out with a picture of coral reefs because it's integrative biology, and we all have to talk about coral reefs at some point or another. So here's a picture of a really diverse ecosystem, and as we know, some are more diverse than others. But in most ecological communities, you can find tens to hundreds to thousands of species all living together in a really small area. So from this coral reef to your own intestine, to the Rocky intertidal ecosystem out here on the Oregon coast. So a central question for ecologists, no matter what ecosystem you're looking at, is how do all these species manage to live together? Why are they there, and do they persist living together? And this question, of long-standing interest to ecologists is becoming more pressing with ongoing climate change. Now, physical scientists are able to take observations of changes in temperature or other climate variables and predict what's going to happen around the globe in specific locations. And this information is incredibly valuable, right? We want this kind of information to get ready for what's going to happen with climate change. So the grand question for ecology is, can we do the same? Can we take what we know about ecological systems now, these communities, and predict what's going to happen in the future? What species are going to be there? What our community is going to look like? To answer that question, we really have to get back to the question of why are species where they are, which is really a non-trivial question. This early drawing by Humboldt shows how many ecologists and scientists in general think about the drivers of where species live. So species are found in places that they can tolerate the climate. So species are found in more cold areas or warmer areas, depending on whether they can tolerate the colder or warmer temperatures, for example. Now, if this is really the only thing that drives where you find species, right, then we're going to be fine in trying to make predictions about climate change, because we already know, we already have a lot of tools for making these predictions, for going from how do individual species respond to the environment now. All we have to do is ramp up the climate in our predictions, and there we go. For example, you could do a climate change experiment, where you take a species, subject it to some experimental climate scenario, warmer temperatures, and see what happens to that species. Or, using observations, you can use an approach called climate envelope modeling, where you look at where species are now, how the climate might change in that area, and predict where they might move with that change in climate. So if this is really all we need to do, then the only struggle is figuring out how to do all these experiments or build all these models for the thousands of species we're interested in. That's sort of a problem. But the bigger problem is that these approaches don't really work when considering how groups of species or communities respond to climate. And this is because species don't exist in isolation. And the interactions among species are really important for determining where they can live. So in scaling that to making climate change predictions, I'll run you through an example. So say you have this butterfly species, you do a climate change experiment, and it turns out they're going to be fine. They, say, aren't really affected by the warmer temperatures, or they can adapt. But if that species relies on another species very heavily, so it relies on this plant for food or for laying its eggs, we also are going to need to know how this species responds to climate change. Now, if it doesn't do that well, say it goes extinct, then this butterfly might also go extinct. And from our original experiment, we didn't think that the species would be that strongly affected by climate change. But in considering its interactions with other species, it may be. So not only do we need to know how individual species respond to climate change, but also need to know their interactions with other species. So this is a really non-trivial question, again, because we're talking about the hundreds, thousands of interactions among species in natural ecosystems. So here are 3 networks depicting the interactions in the rocky intertidal of Chile. So if species are these little bubbles, and their interactions are the lines, then on this left plot we see all of the feeding interactions, we call those trophic interactions, and on the right all of the non-feeding interactions. So links represent who's a predator and who's a prey, or which 2 species are competing with each other, or which 2 species have a positive influence on one another. And this is an incredible amount of work to get this kind of information. Decades of research, dozens of researchers trying to put this information together. So if we really want to go from predicting the response of individual species to climate change to the entire community response to climate change, how do we do that? And I think more broadly this relates to fundamental questions for all science, because we're interested in scaling between fundamental units and the emergent system. For example, in ecology, right, we're trying to go from species to community. But the fundamental unit in other areas of science are particles in physics or genes in systems biology. So the question is, how do we go from here to here? As I mentioned, we do have some of the tools to make these predictions about individual species, right? Those experiments, those models, we can do one at a time. But how do we go from individual species to groups of similar species, or a functional group? How do we go from those to groups of species that interact, and ultimately to this entire community? And are the predictions that we can make about how each one of these levels responds to climate change, are those equally predictable or not? And so this is the question that's really driven my dissertation research, and it relates to an old and foundational question in ecology about whether or not communities are predictable. Can we predict their composition, their response to climate change, or just change in general? And ecologists have really different ideas about whether or not communities are predictable. And so I think that one of the biggest challenges we have is trying to understand, well, given the theory that we have, can we make these predictions, or is that sort of just futile completely? So I'm going to talk today about how my research fits into each one of these levels I explained. First, I'm going to show you how we can move from making predictions about individual species' response to global change to a group of species. And I looked at 5 different species of coral and algae to see how they might respond to ocean acidification, which is a consequence of global climate change. I then looked at how these coral and algae might be important for the community and examined the consequence of species interactions in a low intertidal community. And then, given that these interactions might be important, I'm going to examine a way that you might be able to estimate all the interactions in a community. And finally, see if we can understand how communities change without having to understand every single interaction. And as I mentioned, I've done all this research in the rocky intertidal, so I'll just familiarize you with that system. Here is a picture of the intertidal at low tide. So when the ocean sort of rolls back, for a few brief hours, you can walk right on down, maybe scramble down a cliff, and get to the intertidal. And here, at the very edge of the water, when the tide is at its lowest, is the low intertidal zone. This is incredibly diverse array of species of algae and invertebrates. And over my years in the intertidal, I've seen about 200 different species of algae and invertebrates in Oregon and Northern California. And I went back and double checked, and how many ones I wasn't able to identify, and it was about a third. So I think that this system is really incredible in its diversity, because I wasn't able to identify them, and I think I'm okay at identifying algae. So instead of focusing on these unknown species, I'm going to talk instead about a couple of main players in this system. We have this species of surfgrass. It's the only angiosperm in the intertidal. This is Philospatix. And along with another species, Saccharina, these 2 species form canopies, which means they overlay a diverse understory of mostly algae. So these different kinds of shrub-like algae. I like to think of that as sort of an intertidal forest. We've got the canopy and the little species that live underneath. So we've got different types of understory algae, encrusting algae, mobile invertebrates like the sea star, sessile invertebrates like this mussel. And the intertidal has been a cauldron for community ecology and for the idea that species interactions are important to communities. Because of the small size of organisms, we can go out and do experiments relatively easy though getting up at 2 a.m. is not easy, and using a drill to drill these experiments in the intertidal is not easy, we still have the ability, the capacity to make these quantitative, to quantify species interactions. In the US Pacific Northwest, and in Oregon in particular, here is a fascinating place to study communities, because differences in regional oceanography, here it's shown as differences in temperature along the coastline, drive differences in what the communities look like along the coast. So for example, here in the north, we have a low intertidal community, like the one I just showed you, dominated by algae. But just south of there, in a different region, we have a community in the low zone that's dominated by mobile and sessile invertebrates. This is a really dramatic difference in what these communities look like, and it's driven by these differences in these regional oceanographic conditions. So here we have a really great system in which to study, well, how important are species interactions, how important are environmental conditions for determining which species are living where. So first, in going from predicting individual species to group response to climate change, so as I mentioned before, one of the tools we have to make predictions about species response to climate change is doing an experiment. And here I've plotted the number of different species for which an experiment has been done to estimate their response to ocean acidification. So ocean acidification, again, one of these major consequences of climate change in the ocean. And over the past 25 years, we've characterized the response of almost 200 species, which has been an incredible amount of work. But given the number of species in the ocean, we really don't have the kind of time needed to get to 200,000 species, given the rate of experiments we're doing. And another problem on top of this is that for the species that we are able to do experiments with, they often have a different response. They each have a different response to climate change. So for example, here, if this is some climate change gradient, say it's temperature or precipitation or just sort of abstract change, and a measure of how a species responds to that change in the environment. Each one of these species has a negative response to climate change, but they're all different. And we're interested in, maybe some of these species are more vulnerable, they're going to go extinct more quickly, and some species are less vulnerable. Do we have to do an experiment on every single one of these species to see where they fall along this line, or along this graph? Or is there some common predictor that might illuminate the trend, right? So if we can figure out what species have this red trait or this blue trait, then maybe we could figure out how all species will respond. And so it's really in finding, say, maybe it's larger organisms that are more effective than smaller ones, finding that predictor there. So in the rocky intertidal, we have a system where there are a lot of different species that are likely to be affected by ocean acidification. And here I'm showing you a picture of coral and algae. So they calcify, they're made of the same structure that shells, mussels, clams are made out of. And in this picture, I'm showing was actually 3 different species of coral and algae. So it's a little hard to see, and there's one species here, there's a different one over here, and a different one up here. So as you can tell, they look very similar, they're very closely related to one another, and they're all vulnerable to ocean acidification. So the question is, do we have to study each one of these species on its own to figure out how it responds, or are there some generalities? To do this, we conducted a series of experiments and built a suite of hypotheses based on ecological theory to try to figure out how these different species respond to climate change. We're looking for the commonalities among these species that might predict their common response, rather than what makes these species different from one another. So in their response to environment or global change, how do we predict species physiological performance? It's been long known that the size and shape of organisms can affect how their metabolism, their physiology, so that might be an important predictor of their response to ocean acidification, and we tested 5 different measurements of body size and shape. Or as I mentioned earlier, some ecologists are predicting how a species responds to climate change based off of where it's found now. So we also tested that. And finally, more closely related species might have a more similar response to climate change. But in the end, these 5 species all had a common response to ocean acidification as predicted by their individual body size. So how big this coral and algae was the most important factor in understanding response to ocean acidification. And all 5 species, instead of responding differently, had this common response. So it's a really big step forward in solving this species problem, and solving the problem of having to do a bunch of individual experiments. And if we can just find the right predictor that can, illuminate this common response, then we're going to be able to move from doing individual species predictions to group predictions to climate change. So, given that these coral and algae are vulnerable to ocean acidification, another question might be, well, how important are they? If these species maybe go extinct in the context of ocean acidification, how does the group of species that they interact with, how might they respond as well? So I looked at this experimentally to look at the species interactions in the low intertidal with a specific focus on coral and algae. Just focusing on how these interactions are important for the coexistence or the sort of diversity of the entire system. So here's that picture of the intertidal, again, the low zone. As I mentioned before, there are these canopy species. This is the kelp saccharina, and it overlays the understory that includes coral and algae here and here. And just like other canopy and understory systems, like tree canopies or the kelp forests in the subtidal, we're interested in how these components of the ecosystem interact with one another. Are they shading each other out? Are they competing for resources? Do they have a positive effect on one another? But in the intertidal, it's easy to do these kinds of interaction experiments. You don't need a chainsaw or anything like that. So the main question here, how important are interactions in allowing the canopy and the understory to coexist or live together? And are their interactions always the same? These two questions are really at the crux of community predictability, right? So if interactions are important for determining where a species live, then we need to know that kind of information, and using those single species approaches to make predictions about climate change just won't work. And then the second piece, if these interactions are flexible, if they're change in different environmental conditions, that adds another layer of complexity on top of this whole question about predicting communities. So specifically in this system, I predicted that the canopy with the understory species, they would have negative interactions. They're all algae, they are likely to compete for light and nutrients, and even compete with sessile invertebrates for space. But in previous removal experiments, when the canopy alone has been removed, it always returns to dominance, suggesting it might be a dominant competitor in the system. It might outcompete all these other species and have the strongest negative effects on these species. But it's also been shown in ecosystems that interactions do change, and when conditions are more stressful, species interact in a more positive way. For example, this same kelp saccharina, protects understory species at low tide when it gets hot and dry. It can protect these species like this dark chitin here from desiccating. So in more stressful conditions, the canopy may have a positive effect on the species that live below the canopy. And we tested these hypotheses by comparing a control plot with a canopy removal plot to examine how the canopy affects the understories, and then comparing the same control plot to an understory removal plot to quantify the effect of the understory on the canopy. And this experiment was replicated multiple times, first at 10 sites along the Oregon and Northern California coasts, again because these different sites are different in their oceanographic conditions, so maybe the interaction would change along this coastline. And then a second time, the experiment was replicated at 3 different levels of wave exposure, so thinking that wave exposure might shift these interactions between negative and positive. But it didn't really matter. So the interactions were actually stable, the same across all these different conditions. And unlike the prediction that the canopy would have the strongest negative effect on the understory, we actually saw the opposite. So for example, here's an understory removal plot early in the experiment. The canopy here is at the center. And that same plot, by the end of the experiment, the canopy was completely lost. There's just this root-like structure, and the canopy can't regrow from that. And this was a general result. So the strongest effect in this, or the strongest interaction in this system, was the effect of the understory on the canopy, here shown as a positive interaction estimate. Now the canopy had mostly neutral effects on the understory, though some positive and negative effects. By far, the strongest effect was that of the understory on the canopy, and so to really sort of untangle what that's being driven by, we did a series of surveys where we looked to see where the canopy juveniles were found. So in all these different components of the understory, these canopy juveniles were found growing exclusively, almost exclusively, on these coral and turf algae by an order of magnitude. So it really seems that it's the coral and turf it's specifically the coral and turf that facilitates the canopy. So putting this all together, encouragingly for predictability, these interactions were stable, right? They didn't change in different environmental conditions. That would really have thrown a wrench into this system. But we found that the canopy had mostly weak effects on the understory, the positive effect on these crustose algae, and a negative effect on these turf algae. Interestingly, which you can't see, there's an arrow that goes there to show that there's no effect on coralline algae. So it has a negative effect on the turf, but none on the coralline. And these coralline algae, in turn, have a positive effect on both the adult canopy and the canopy juveniles. So, getting back to this original question, what would happen if the corallines were lost from this system? Well, we can sort of run that hypothesis through this interaction web, right? Coralline algae are lost because they have such a strong positive effect on these canopy species. Those species might as well be lost. And they have positive effects on species that live below it, so those species might not be able to persist without the canopy. And the species that is negatively affected by the canopy, once the canopy is gone, might come to dominate. So if you were going to try to make a single species prediction about the impact of climate change, global change on this one species here, you would have made a really different prediction than what might unfold, given the fact that this species so heavily relies on coralline algae, which are vulnerable to ocean acidification. So through this experiment with just a couple of interacting species, we're able to quantify what the interactions are, quantify their consequence, so what happens when those species are removed from the system, and make some predictions about how the community might respond in the context of at least ocean acidification. So, here, species interactions are important in this community, right? How do we move from these species interaction experiments to estimating the importance or the impact of species in an entire community? Like I mentioned, the intertidal is a really easy place, seemingly easy to do interaction experiments, but they've quickly become intractable, right? So there's dozens, hundreds of different species in the intertidal. How do you do field experiments looking at all of those interactions? And in some ecosystems, like this tropical rainforest, it's just not going to be possible to quantify every interaction among every species. So how do you do this? Is there a way around the interaction experiment? For a long time, ecologists have been toying with the idea that species interactions can be inferred from observations of where species are found occurring. And I'm going to walk through an example of what that means. So say these two purple squares, each one is a habitat patch. And if they have the same environmental conditions in each habitat patch, they're pretty much the same. And you always find species co-occurring. They're always found together. Then you might think that this is a signal of their positive interaction. That they're that they have such a positive effect on one another, you can never find one species without the other. On the other hand, if this habitat patch, again, is the same habitat in each area, but you never find these two species together, that's been interpreted as a signal of their negative interaction. That they can't be found together because they're competing. So there's a suite of statistical methods that purport to estimate species interactions from these observations of which species are co-occurring. This is really interesting because that's usually the province of interaction experiments, where you look, experimentally control, okay, which species is where, and measure how they respond in the presence or absence of one another. And overall, the results, the interactions inferred from these co-occurrence analyses have never been compared with the results from an interaction experiment. So this is a huge gap. And it's really troubling because there are so many different kinds of statistical co-occurrence methods that are coming out. Here are just a couple of different ones that have been recently published, but we're seeing a lot of new methods to estimate interactions from these co-occurrences, and there's no consensus method. So no one method is considered to be the best. For example, each one of these recent high-profile papers uses a different method to estimate species interactions from their co-occurrences. So a pretty basic question is, among all these different methods, do they detect the same interactions? So what kind of inferences are we drawing based off of just choosing one method or another? And then, do they match what we've measured in experiments? So I went out and surveyed the Rocky intertidal again, focusing on the low intertidal zone and recording the presence of and absence of species along the Oregon coast. Then feeding these data through the 7 different co-occurrence methods I mentioned earlier to gain these predicted interactions from observational data. And then I compared them with species interactions as quantified in the literature through experiments. So the survey show, we got 97 species from the survey. And looking through the literature, I was able to record 586 interactions among those 97 species, which you can see here. So the little gray circles around the edges, each one is a species. The interactions among those species are coded with a red arrow for a negative effect and a blue arrow for a positive effect. Just note that if there's no interaction between 2 species in this figure, it's not because that interaction doesn't exist, but because it maybe hasn't been experimentally measured yet. So this is just the ones I was able to find. But in comparing these interactions as measured by experiments with those from co-occurrence analysis, this is what that looks like. So first, you'll note just visually that among the 7 different co-occurrence methods, they're all generating really different predictions about what species interact and what those interactions look like, whether they're positive or negative. And then comparing each one of these to the experimental interactions, we're not seeing much concordance. I've done some statistical analysis to examine specifically the differences in these different interaction networks, which I don't have time to go into today. But I'll just note that when you compare each one of these sets of interactions as estimated by co-occurrence analysis, and compare it to how many pairs of species it matches in the experimental database, it matches between 0 and 4% of the time. So that's a really, really low level of matching between what we estimate through interactions and what's estimated by co-occurrence analysis. You also note that there are some of these networks that you really can't tell what's going on there. There's so many different links that it's hard to tell, well, maybe they are estimating some of the right ones, right? Maybe you just have to estimate a ton and then you'll get some of the right ones in the end. So another way to look at this is we have the number of interacting species, and we're going to look at the relationship between the number of species in the network and the number of interactions among those species. Note that this is on the log scale, right? So, and the gray line, the gray dotted line at the top, is the maximum number of interactions you can find among the 97 species in the data. So that's just if every species interacted with every single other species. And this red line is what we might expect the relationship between the number of interacting species and the number of interactions among those species to be, just from previous data. So mostly these relationships have been studied for food webs, and I'm talking about non-trophic or non-feeding interactions, but this is our baseline expectation. And in plotting where the co-occurrence methods fall along this pattern, we see that those 3 methods with the really dense number of interactions fall out up here, 3 orders of magnitude higher than we expected, than really has been measured in any other system. So the ill correspondence between both among the different co-occurrence analysis methods and between them and experimentally estimated interactions really calls into question the way we're using these methods. So that's a big problem, right? And in knowing the species interactions, it doesn't mean that we can estimate their co-occurrence or vice versa. So there's this real disconnect here between trying to infer a process from a pattern, right? And again, I think that this is a question that is more broad than just thinking about species interactions and co-occurrence. It's about how much we can learn from a process just by observing the patterns that process maybe produces. So we start to reach the limits of predictability here, right? We know species interactions are important, but we're running low on ways to estimate those interactions. And that really brings us down to a sad note. But there's still some way forward, right? And my last chapter focuses on how we can get at understanding how an entire community changes without having to estimate and track every single interaction in the community. So is there some predictability left in that? And one way we can do this is by examining a community's response to change, to a disturbance. It's really the ultimate test of predictability if you disturb a system and see whether it comes back to its original state or it goes to a completely new and alternate state. So what might make a community return to its original state? I think in a really simple way, you could just consider what species are living nearby and whether they have the capacity to regrow or disperse back into that spot. So for example, in the low intertidal, we have the surfgrass Philospadics and these turf algae. They have the capacity to regrow. They have crust, or the Philospadics has a rhizome, and they can just grow right back into a disturbed clearance. And Saccharina, the kelp canopy I talked about earlier, can recruit back in. I mean, it might depend on one or another species, but it can recruit. So if these species are nearby a disturbance, we might expect that they could just easily recover. And a way we might visualize this change is with an ordination. So here I have a non-metric multi-dimensional scaling plot, mouthful, but it's just a simple two-dimensional way to visualize how similar communities are to one another. So here I have a group of communities, little C's. The ones that are closest to each other are most similar in what species are there, and the ones that are further are less similar. And we can visualize how these communities change after a disturbance by tracking what species show up in the little S's and where this sort of community space goes. So they're becoming less similar to their starting place and then more similar by the end of the time period we're observing them. And one might expect that communities that start out really similar and are subjected to similar environmental conditions might all end up back in the same spot. But there's a lot of reasons we might not see that such a easy recovery, right? For example, here's a disturbed area in a Philospadics plot. You can see that Philospadics right around the edges here. And right in the middle of this disturbance are a bunch of baby algae growing. If you're not as obsessed with algae as I am, you might not realize that a lot of these species growing in here are species that aren't found in the intertidal. They're ones that are found in the subtidal, or ones that can only live underwater all the time. But after this disturbance, all of these new baby subtidal algae showed up. And so in marine systems, because larvae, spores, seeds float around in the water, they're able to colonize open space very rapidly, and often when they colonize this space, they can just grow and grow and grow, take over, and the community becomes more different than we would have expected just based off of who lived nearby, right? Because things can disperse. And the way we might visualize that is, so we have our communities in this return to communities, the same community in the beginning. And if we have these recruitment events, or these new species colonizing space, they might end up in really different places than you would have predicted. So to look at how predictable communities are based off of who lives nearby, I conducted a 2-year recovery experiment in the intertidal, clearing out these experimental plots of all organisms and tracking who showed up over 2 years. Excuse me. And specifically clearing these clearances, or these experimental plots, in 3 different areas of the intertidal. So we can take advantage of the fact that there are these huge patches that are mostly dominated by Philospadics, Saccharina, or algal turfs. And within the patch, making a clearance, we can make predictions about which one of these is going to return to its original state. As I said, Philospadex has the capacity to regrow, and it's also a competitive dominant, so it just overgrows everything else in the system, and that's been shown in previous experiments. So we expect any disturbance in this Philospadex area to become Philospadex. Similarly with Saccharina, as I said again earlier, when you remove Saccharina, it always comes back. And those have been experiments that were done back in the 70s, but it really depends on whether or not these coralline algae are present, which is what I showed as an earlier chapter of mine. Now finally, turf algae, again, they have the capacity to regrow. They have these crustose forms that can sort of regrow back into a disturbance. But unlike Philospadex, where there's one species, it's the dominant, it's always going to dominate, in turf algae, we think that the interactions are much less structured in the way that there's no one species that's a better competitor than the other. So maybe you might get back a community of turf, it just might look a little different. So in doing these experimental clearances in these broader patches in the intertidal, I also replicated this experiment at 6 sites along the coast. Again, with sites differing in their regional oceanography, so the question being, do these communities all return? Are they very predictable along this entire spatial gradient or not? So first, you can see, we started out with the experiment here in spring 2013. All these communities are really close to one another at the beginning of the experiment. This is before I did the clearance. And after the disturbance, they really spiraled outward, and they changed very rapidly. And you can track what happened at each site, depending on the different colors here. But that's almost not as interesting to me as the fact that they're all moving in the same direction, right? That some sites, by the end of the experiment, here in these triangles, are more similar to the beginning than others, but they're all really following that same trajectory, that same channel of recovery back towards the original state. And we see a similar pattern when we look at these experimental plots that I cleared in the Saccharina or the turf bed as well. It's a little less clear. But overall, 50% of the plots return to their original state after 2 years, which I think is sort of remarkable, just because we do see these colonization events that can be pretty random. We still see that 50% of the time they recover. And among all of these algae, even I had a hard time telling that that was an experimental plot after 2 years. So, like you could see in those ordinations, these 3 community types really dominated again by the end of the experiment. But we also saw a new community emerge. So sometimes a community in one of these Philospadex, Saccharina, or algal turf areas became dominated by sessile and mobile invertebrates, which wasn't what we expected at the beginning of the experiment, right? We're making disturbances in these areas. We didn't necessarily think that it would become dominated by invertebrates. But if you remember back to the beginning of the experiment, I noted that in some areas of the Oregon coast, macroalgae are favored, and in some areas, invertebrates are favored. And so when we found these new invertebrate-dominated communities, it only happened in these regions where invertebrates recruit very highly and grow very quickly. So even though this new community type was unpredictable, there's still some predictability in the unpredictability. And to break down which of these experimental plots, sort of what community they turned into, we only ever found plots that were cleared in the Philospadex bed turn into Philospadex. So that really matches that original prediction, right? That Philospadex, dominant competitor, great at regrowing. They sometimes became turfs, but really mostly Philospadex. Plots cleared in the Saccharina, the kelp bed, also became Saccharina, but equivalent with the number of times that it became turf or invertebrates. And these plots in the turf bed also became both invertebrate and turf dominated. To me, the surprising piece here was Saccharina, right? That it was equivalently turned back into a Saccharina community or a turf or invertebrate community. But even among, again, that unpredictability, we find that there are some things that we can make predictions about. Because of the 75 times you find a new Saccharina colonizing, 69 of those times, coralline turf algae was present in the community in one of the time steps before. which matches onto the earlier work I did showing that you need this species for Saccharina to show up. And by the end of the experiment, just knowing what kind of species was around a disturbed plot, so either is it a Philospatix community, a Saccharina one, or a turf one, just knowing that and what site along the coast the experiment was conducted at, we could explain 60% of the variation in community structure. And I think that that's really a lot, considering how diverse this community is, how unpredictable we think that the process of colonization is. We can explain 60% of the variation in who's there. So in thinking about can we make predictions of entire communities based off of species interactions, that might be a difficult task. But in coming at it from another direction, in trying to scale back and not track every individual interaction in the system, we're able to gain back some predictability. Because communities didn't become pretty much any possible community on the planet. We didn't see ones that were so novel they've never been seen before. After disturbance, we saw the same communities that were nearby a plot, or one that is pretty characteristic of that region. So returning back to the beginning of the talk, where have we come in trying to understand the processes that drive the structure of diverse communities, and can we make predictions about what they're going to look like? I've shown that we can really gain predictability at all of these different scales. So by using ecological theory and building individual hypotheses and testing those experimentally, we can make predictions about groups of similar species, which is a huge step forward in dealing with the consequences of climate change for diverse communities. And then I showed that by doing an experiment, you can quantify the interactions among a small set of species, their consequences, so what do interactions mean for this community. And though it might be difficult to estimate the interactions for an entire community, there's still some power that we have to make predictions about how that community might change. Specifically in the intertidal, we've learned a lot about coral and algae and its interactions, right? So these coral and algae facilitate important species, which in turn facilitate other species. So I think in trying to understand what species are most vulnerable to global change, and then understanding their interactions, we can really get there in these smaller sort of components of the community. And if we're trying to get between individual species and entire communities, we have a lot of different approaches to do this, and I think we need to use them all. Because in the end, I don't think we can really get away with not understanding, quantifying species interactions if we want to make predictions about communities. It's just that we might not be able to make all the predictions we want to. We might not ever be able to understand all of the interactions in an entire community, an entire ecosystem. And instead, we might have to scale back our expectations and focus, again, on these vulnerable species. How do they affect the community? But I also think that we can put our heads together and think about innovative and cool ways to measure species interactions. We have the capacity to do interaction experiments in some systems, and these co-occurrence methods, for example, I think were a really great attempt at trying to understand interactions. We're still working on what those mean, but in the meanwhile, there are some people who are doing really using cool new methods like remote sensing to detect positive interactions, or automated video analysis to detect what species are feeding on which. So I think that there's a lot of capacity to use new technology to measure species interactions. And in the meanwhile, while we're sort of plugging away at this problem, I think there's a lot of power also in taking a step back. Again, we might not be able to make all the predictions we want. We might not be able to say, okay, this community will specifically become this one with these exact species. And we have a lot of tools to make more general predictions. For example, macroecological theory makes predictions not necessarily about the composition of species in a community, but about the number of species in a community. Because things like species richness, or the number of species in a community, that's a property that's highly constrained. So, and it can be predicted from sort of the first principles of ecology and chemistry and evolution. So, in the end, sometimes we want to make predictions about everything. And I think it's more important to focus on when we can or cannot make predictions, rather than trying to universally make a prediction about how every community might change with climate change. because I don't know that this is a question that I or anybody can really answer. But we can take a step back, a 10,000 foot or a couple hundred foot view, and think about, again, where is our predictability constrained and where can it be developed? Because even facing an uncertain future with climate change, we have the capacity to understand how individual groups and small communities of species might be affected by climate change. And in using and developing ecological theory, we can make more general predictions about how ecosystems might respond to climate change. So, with that, I'm going to do a couple of acknowledgements. And I first want to thank all of the collaborators and co-authors on these papers, which this said Kyle Koblenz, but it just, I don't know what happened to my talk. Kyle, wherever you are, I'm sorry. An incredible group of people to work with. Of course, Sally and Bruce I'll acknowledge in a second, but I just want to call out Francis here, who's been an incredible mentor, and I've really enjoyed all of our work together. This work really wouldn't have been possible without the generous funding from a lot of different places. I want to specifically point out a graduate research fellowship from the EPA, which supported me for about half the time in school here, and one from the Hatfield Marine Science Center, which supported the work for all the coralline and ocean acidification questions. So much support from both the IB office and from the PSCO office, I guess. So Cindy and Kathleen have always been amazing, and Mike with all my data and questions about how to use Fortran, which didn't go well. My committee, Mark, Alex, Scott, and Selena, thank you so much. Mark, so many questions over the years, but I mean, I'm so grateful. This is my picture, which you can only barely see. It's this old-timey picture of Bruce and Sally we took a couple years ago with the poorly developed film from an automatic camera. They've been such incredible mentors. I mean, they mentioned how much I've been out in the field, and what they didn't mention was how they were out in the field every single day with me. Here's a picture of Sally, incredibly spending the hours and hours after one morning in the tide, building these little communities. We chiseled them off the rock, glued them back together in an experiment in my first year that never went anywhere, and I never really collected any data for. So I just I want to highlight all this work that they put in to help in the field, in the lab, everywhere. Bruce, all of the years of pointing things out and advising, all of the years of movie nights and with our cool 3D glasses. Because I know it's not easy to work with me in the field when I haven't had enough coffee. But I've had an amazing field team, and I don't know, I'm so grateful. I really want to point out here, this is Kelsey, she's an undergraduate who worked with me on the coral and algae project, but also helped with so many other components of my dissertation, just happily plugging away, counting corallines. Here's Jessie, who, wherever you are, I did not forget that those days you were out with me recording for that co-occurrence chapter so many years ago. Oh, and I can't forget, I really wanted to thank Jared, because this is the best picture I could find at the last second of him, who up until this weekend was sending me funny Photoshop pictures to make me laugh because I was stressed out, so amazing. A group of amazing people who I didn't necessarily do my dissertation work with, but so much other inspiration. Kirsten, Annalise, Jane, I admire you so much, and it's been a really great time working with all of you, and I really hope that we keep working together in the future. My broader lab community, it's again been such a great time, and I know we'll be friends not just outside of the field, but continuing forward. The IB department, the graduate students here have been such a fun crowd to spend time with, and I can't believe it's been 6 years. Specifically in the last year, I know I've been a bit of a handful. It's been a little stressful finishing up. So I want to have specifically call out this group of people who've been so supportive and amazing this last year. Sheila, Leah, Kyle, Jeremy, Caleb. It's been, I rely on you so much. And I wouldn't be anywhere if I weren't going to mention Liz and Chen Chen, who started their degree program in the same lab, in the Mengi lab as me at the same time. We lived together, did everything together. And I picked this picture out because I look taller than them. Which Liz, I'm sure, will point out that I am not. But I absolutely could not have done this dissertation without you two being by my side the entire time. And of course, my family who traveled all the way here from Connecticut and New York, my brother who took off time from doing his master's thesis, which I cannot believe you came out. It's amazing. My sister who saved up all her vacation time to come out for a couple of days. You guys are amazing. So with that, I'll take any questions that you have.
Great talk, Allie.
Thanks.
Really enjoyed it. Can you talk a little bit more about the mechanism by which the coralline algae facilitates the overstory?
Yeah, I think it's a really interesting question, and it deserves some more experimentation, but we were able to come up with a few ideas. So as I mentioned, the understory corallines, they affect both the adult canopy and the recruitment of the understory. I think that the understory coralline algae might protect the canopy at low tide from drying out. So these corallines actually hold more water than other understory species, so they sort of are a little moister, a little wetter when the tide goes out. And Saccharina, the blades lay over that. And so often we saw that the blades came off of the rock first and then the holdfast, so I wonder if that's maybe the mechanism. And then for the recruitment, I think it's potentially some protection from herbivory, because the corallines, nothing really eats them, and those spores can just sort of rest out. It's a nice refuge. They can get all happy, grow old, and pop up there. So I think that there's a little more work to be done to figure exactly what that mechanism is, but those are our ideas so far. Yeah, Lillian.
I'm interested in the last bit where you were doing the clearance
Mm-hmm.
and then tracking the communities for 2 years.
Mm-hmm.
And the type of disturbance is kind of interesting
Mm-hmm.
because you were just clearing a patch and then I assume not clearing it again, but
Mm-hmm.
tracking its regrowth.
Mm-hmm.
And how that might be different than something like climate change or acidification that are
Mm-hmm.
more,
Mm-hmm.
it's a continuing disturbance.
Yeah.
And so you might not
Mhm.
predict that you have the opportunity
Mhm.
to
Mhm.
return, and how you
Yeah.
think that kind of disturbance regime would give you different results potentially.
Yeah, so I guess I think about it a little bit differently, that those communities, it wasn't that I was exactly trying to use that disturbance as an analog of climate change, but instead sort of utilize the opportunity of the disturbance to look at how these communities responded to perturbation. So how persistent are these communities? Do they go back to their original state? Because as you said, a disturbance from climate change is going to be a pressed disturbance. It's going to happen slowly, gradually through time, maybe a couple of extreme events. These disturbances, it's a pretty traditional way to do a disturbance in the intertidal, because we scrape everything down, scrub it down, bleach it, scrub it again, bleach it, scrub it again, then flame torch it. So it's like not exactly a realistic disturbance. But instead it's just, okay, if we start from zero, we clear everything out, what comes back? I think with climate change, we're going to see the gradual loss of certain species, maybe ones that calcify like these coralline algae, but also a lot of the invertebrates. Or with changes in temperature, maybe it's other species that are more sensitive. We don't have a good idea of which one of these algae species are sensitive to changes in temperature. So it's a really different, those are two different ways to think about changing communities, but I think they can be complementary. Yeah, Will.
How do you think different communities will respond to something like ocean acidification, where you have kind of algal-dominated communities where they're able to kind of absorb some of the carbon dioxide in water, maybe buffer it a little more versus an invertebrate-dominated community where potentially they don't have that same mechanism?
Yeah.
Do you think that's going to play a role in how communities change in time?
Yeah, I feel like I could talk about this for a really long time. It's so interesting, and we're only getting to that level of making these predictions about communities. Because that first experiment that I showed you with the groups of coralline algae, I didn't talk much about the specific results, but we found that there's this link between coralline algae, their calcification, and their photosynthesis. And that individual corallines that can photosynthesize more have the capacity to buffer themselves from the detrimental effects of ocean acidification on calcification. So algal communities, when they're all mixed together like this, where some species calcify and some don't, they might have this response to ocean acidification that's difficult to predict because we don't know that much about photosynthesis in all these species. I think as a really general prediction, you might say that these invertebrate-dominated communities will be more affected than these algal-dominated ones, but there's still a lot to learn about how individual and groups of species respond to ocean acidification. Specifically in this system, they're exposed to really acidic conditions on a regular basis, so they might be sort of locally or pre-adapted to those changes. Jean.
Very nice job, Allie. I'm curious about your initial overall framing of ocean acidification as a subset or consequence, you said, of global climate change. Because that's not the way I think of it. I think of carbon dioxide causing climate change, excess carbon dioxide causing ocean acidification, so they're parallel problems.
Yeah.
It's not that OA is subsumed or a consequence of climate change.
Yeah, I mean, I think you could frame it that way as well. I think I was just trying to distinguish it from the climate change as its own process. You know, most people are thinking about climate change, they're thinking about the terrestrial ecosystems, changes in temperature, precipitation, and whatnot. And I was just trying to distinguish the two. But yeah, they fit together more as a consequence, a joint consequence of changes in greenhouse gas. So.
And for sure, all the consequences of climate change, many of them affect intertidal communities, and OA is also affected.
Yeah.
But one of the reasons that OA hasn't gotten the attention that it has is that I don't think people have really framed it as a problem that is equivalent to climate change.
Yeah, I mean, I think that we're sort of catching up with the work that people have done in terrestrial ecosystems on climate change, and even in marine ecosystems on changes in temperature. That work that I showed you of all the interaction or all the single species experiments, you know, there's some work done back in the 70s, and we're really just rapidly trying to catch up to what we know about ecosystems on land. So yeah, there's a lot of work to do. Yeah, Adam.
So I thought your comparison of the different co-occurrence models was like really cool and really important. Actually, it's kind of a reality check of how realistic are these things.
Yeah.
So I kind of have a two-part question, knowing nothing about any of these methods.
Okay.
Right? But not only is the question to me kind of which of these methods are best, but how much room is there kind of within models for different kind of parameter optimization, right?
Yeah.
And then can you use those empirical data sets as a way to kind of pick the best model and then train the best model?
Yeah, that is a great question. So within each one of these co-occurrence methods, let me flip back to them just so that you can see what I'm talking about. Within each one of these, there is a lot of flexibility for making changes, right? And so some of these methods, like this checkerboard pair method, it uses a metric of species co-occurrences. So you could pick from a suite of, I don't even know now, like 20 different metrics to make these predictions. For correlation, you can use whatever correlation metric you're interested in. Some of these methods, this JSDM, this joint species distribution residual, that's a Bayesian model that you can change a lot of the components of. But I think, and I've played around with a lot of these, and I'm mostly focused on, well, how have they been used in the original paper that was published? You know, what are the sort of default parameters, and that's what I focused on because I'm making so many comparisons. And from this work, I honestly haven't attempted to try to improve any one of these methods. That was really where I started my PhD, because I was like, okay, great, I'll do an experiment with the interactions, and then I'll just estimate the rest with one of these methods, that's great. But in thinking about it over the past 5-6 years, I just don't think that they're translatable. And so working on improving any single one of these, I don't know if that's heading us into the right direction. And there are people who are doing that, and I would be really interested to see where they go. But I'm more interested in trying to, in maybe like closing this area off. Like, what can we learn from this, and then are there other ways to get there? Oh, yeah, Becca.
My question is also about
Yeah.
these characteristics and
Yeah.
playing around with too, so I find it really interesting.
Mhm.
And one of the things that they suffer from is habitat filtering, because even if you have two species that are always aggregated,
Yeah.
they're bringing the same habitat,
Yeah.
they interact with
Yep.
they're different like that.
Yep.
So if you were to, I don't know if there's analysis at all, but if you somehow able to pull out a habitat, because you get more concordance with yours, where I'm assuming you're actually taking that into account.
Yeah, I'm really glad you asked that question. I don't have those pictures. I did do that though, yeah. So I accounted for the effect of the environment using, I think, 13 different environmental variables. It's just as you said, maybe they're always found together because they do really well in that habitat, and it doesn't have anything to do with their interaction. So I accounted for, what site they were at, what side of the rock they were on, sort of like the slope of the rock, how wavy that area was, what the temperature was, all the different environmental variables that I could come up with that we had data for the intertidal, and threw those into all of these models. Not all of them can specifically, in and of themselves, account for the environment, but there have been a couple of published papers that shows how you can do it after the fact, sort of subtract the effect of the environment, and then see what's left. And they don't make this any better. It doesn't improve our predictability here, which was really surprising to me because that's really becoming more and more common. People are trying to remove the effect of the environment and just focus on the interactions, but it doesn't seem to work. Yeah, Kevin.
Sorry if this is a basic question, but what is the goal of making a community prediction? Or like, what does that look like? Is it like a species list for after some effect happens, or is it a few particular species you're interested in, or is it this will be Orland dominated or not?
Yeah, I think that's one of the ways that we could start improving this kind of research is focusing on what kind of prediction we want to make. I was thinking about what is a pretty common method of making a prediction about an individual species, where it's going to go, so say we have like this X mark location, this is one we're really interested in seeing who's going to be there. Well, you make a bunch of these individual predictions, and you sort of stack them together. You add them all up, and you say, okay, well in this like X location, we're going to see, we now see these 2 species, in the future we'll see these 5 species. So that's called stacked species distribution modeling. People are using that more and more and more often. And so, yeah, a lot of people are interested in this one location. What species are there? How abundant will they be? That sort of thing. And maybe, again, those are the kinds of predictions that we might not be able to make, but it could be, well, these are the species that could be in that region, or these are the types of species that might be around. Maybe they are the ones that don't calcify or something like that. But there's a lot to play around with in figuring out exactly what our question is.
One more question? Or no more question?
Oh, yeah.
So kind of along that last question, you looked at how the species composition changed with like biodiversity and like species identities and uniqueness. What about looking at things like productivity and ecosystem functions? Do you think that is changing at all?
With climate change?
Or like just with, somewhat within the intertidal?
Oh, within the intertidal? I haven't worked much on productivity or ecosystem function. I mean, I think that that's really the next level up. And in a lot of ways, that's the level we care about. It's not just, okay, is species A, B, C, and D, are those guys going to be there in the future? But it's sort of what effect do they have on the ecosystem? Is that going to be more or less productive? Is it going to provide more or less habitat to other species? And so I think that it's that link between species identity and ecosystem and function and productivity that's important. I haven't looked at it much. I think, I'd be interested in the future. So, I don't know.
Thanks, Sally. Before we give her her final round of applause, the committee will meet in the Dornfeld Library, I think. And after the committee has met with her, there will be a gathering in the Lutsenko-Mengi lab, which who knows when it'll be exactly, but it could be like 2:30 or so. I'm sure there will be emails sent out, but you're all welcome to join us. Let's thank Ellie. Great job.